More formally, I mean for individual I, we know the probability it has QTL genotype J given its available marker data. And we have this model that the phenotype for individual I given its QTL genotype follows this normal distribution, follows a normal distribution with mean depending on QTL genotype and some common variance. But given the marker data, that we don't have a normal distribution, we have this mixture of normal distributions. We have the normal distribution here for different QTL genotype averages weighted by these probabilities. And we're going to use this probability to estimate the means and the SDs. And we're going to do that in the way that we're going to do that by maximum likelihood. We're going to try to find these three estimates for which the observed data is most probable. And the way we actually do that is using what's called the EM algorithm. The way the EM algorithm, the EM algorithm is an iterative method for finding these, for finding the maximum likelihood estimates in this sort of model. And it has two parts, an E step and an M step. In the E step, what we start with some initial estimates of the two genotype means or the three genotype means in intercross and the residual SD. And we use those initial estimates to calculate the probability that to update sort of our understanding of which genotype group individuals are in given their phenotype data. Previously we'd calculated for each individual what's the chance that it has each possible QTL genotype given its marker data. In this algorithm, I want to calculate a similar sort of probability, but given not just the marker data, but also given the phenotype for that individual. But to use the phenotype to inform me about QTL genotype information, I need to know the effects of the QTL. So I start with some initial estimates of the effects of the QTL, and then I calculate this. This uses Bayes' rule to calculate the probability that an individual is in, update the probabilities associated with the three QTL genotypes, given not just the marker data for that individual, but also its phenotype. I'm going to use those as weights, and then I'm going to estimate the three genotype means by, instead of taking the average phenotype for, I'm going to estimate, say, that the average phenotype for individuals that are in genotype group CC. I'm going to take that as a weighted average. I'm going to take a weighted average weighted by the probability each animal or plant is in that genotype group. And a similar sort of formula for the residual SD that is there for completeness but not worth mentioning. The key idea in this algorithm is that I use some initial estimates of all the parameters to update my understanding of which genotype group an individual is in. And then I use those as weights to get an improved estimate of the QTL or the QTL effects. And then I'm going to repeat these until they stabilize. And I have a little interactive chart to try to illustrate this a bit further. You think, at a given location in the genome, in a backcross. A backcross is easier because I just have two genotypes, And so I can really just think about the probability that you are heterozygous. So at some marker, I have, for those individuals that are homozygous at the marker, there's a very low chance that they're heterozygous at the marker. So on the x-axis here, I have the probability calculated initially that they're heterozygous. So a bunch of individuals that are very likely to be homozygous, and so the probability they're heterozygous is 0. A bunch of individuals that are very likely to be heterozygous, and so their probability of being heterozygous is close to 1. And then a group here and a group here, maybe because they're missing a nearby marker, and so they have a little bit less information. And a group in the middle that are maybe recombinant across the region around the QTL. And so they're either a bit above a half or a bit below a half of are they heterozygous or not. So in sort of the input to QTL analysis is, for this interval mapping method, is these probabilities for each individual and then what phenotype they have. Really, the first thing I do is I get my estimate of the average phenotype for these heterozygotes is to take the weighted average of the phenotypes weighted by this number. And the average phenotype for the homozygotes is a weighted average using basically flipping this, 1 minus this probability. Yeah? The reason they're a bit below is because just if there were just 2 markers and everyone were genotyped there, then everyone would be either really close to 0 or really close to 1 or at 2 spots in between. There would be just 4 groups of animals with 4 different probabilities. But in practice, I have maybe a bunch of markers around it, and they're missing different amounts of data. Depending on which amount of data they're missing, they'll be, shifted a bit.
So this is based on just 3 markers. This is based on
the whole chromosome. So the first step in the EM algorithm, I say, given my current understanding of phenotype averages and given their phenotype, use that to shift my probabilities. My initial estimate of the effect of this QTL is that the homozygote has a slightly higher average than the heterozygote. And so if you're over here and have a high phenotype, I would say you're probably a bit more likely to be homozygote. And if you're down here, have a low phenotype, you're maybe a bit more likely to be homozygote. A bit more likely to be over there. So everybody tilts a little bit. And then I use those new weights, everybody tilts a little bit, and then I use those new weights to get improved estimates of the 2 genotype averages. And then I do the whole thing again. I use those new estimates that are a little, you can see they got a little bit farther apart. And then I recalculate what is the chance that they're homozygous versus heterozygous. Everybody tilts a little bit more. And I adjust those estimates and they get a little bit farther apart. And I repeat this until basically nothing's changing anymore. And then I stop. And the final values there, this thing will converge to the maximum likelihood estimates. Yeah?
So I guess modern variant callers output genotype probabilities as part of the variant calling process. Would there be any benefit to including those genotype probabilities output from, say, when it's call it variance in sequencing data to inform the probabilities estimated here?
Yes, potentially. But RQTL can't use that information. Yep, and that may be especially important in these multi-parent crosses. Whether you're doing genotyping on a microarray or by next-gen sequencing, using the actual counts of the two alternate reads at a SNP, or using the SNP intensities could give you better results. But everything will be slower, I'm sure. His question was, can you, in modern sequencing technologies, you have more information than just a genotype call, but you have some nice information about uncertainties, either in the intensities of the alleles on a SNP array or the actual counts of reads when you're doing sequencing. And can you use that, and should you? And I say, you can in principle, and yes, you should, but not with RQTL at this point. Yeah?
That seems to converge or at least stabilize pretty quickly. Is that typical when using the EM algorithm?
In this QTL mapping context, it will stabilize pretty quickly. Yes. The question was, it looked like it converged really fast. Is that typical? I would say, in general, for the EM algorithm, it's not typical. It's known as a rather slow algorithm. But in practice, in this problem, it works really well. And it has a bunch, the implementation of this algorithm is much easier than most alternatives. And it's more robust than almost any other one in that, like if you did Newton-Raphson to try to optimize the likelihood, that often just goes off to infinity. This thing never does that.
Is that because of a smaller data set, perhaps?
Part of it is that we really have a ton of information about these two parameters. We don't have very many parameters, and we pretty much know what they are. And we'll come back to this in a minute, but really that initial estimate of just use these probabilities and get those averages is pretty close to almost all you need in many cases. So having calculated, obtained those estimates of the effects of the QTL, I calculate this LOD score, which is our measure of evidence for a QTL at a given position. So the LOD score is to calculate the probability of the data if there's one and only one QTL at a given location, and given my final estimates of all these parameters, divided by the probability of the data if there were no QTL at all anywhere, in which case I assume that all the phenotypes just follow some common normal distribution. Take that ratio, the log base 10 of this likelihood ratio, that's the LOD score. And then if for interval mapping, I first calculate all these genotype probabilities everywhere in the genome for all animals or plants, all individuals. And then I consider each of these putative QTL locations one at a time, and I apply this EM algorithm to get estimates of QTL effects and shove them into this formula to calculate this LOD score. I repeat that across the whole genome. Oh, and I forgot, one and only one time I calculate this, the denominator in this ratio, the thing on the bottom. So I calculate all these genotype probabilities. I calculate this thing on the bottom once, and then at each putative QTL position, I'm going to calculate these. I'm going to apply the EM algorithm to estimate these, plug them into this formula, calculate this log curve. And, for these data, I get pictures like this, which we can, I think, calculate on our own right now. So we should have in R, this one object, SUG, that is our cross object. I created some other object X that I removed. If I want to remove an object over time, there's a function RM, parentheses, will remove an object. The first thing I need to do is calculate these QTL genotype probabilities. And the way I do that is with a function called calc.genoprob. It takes, the main argument is this cross object. It has a bunch of other arguments. The most important one is called step, which determines the density of locations at which you're going to calculate these probabilities. So if I say step equals 1, it will calculate QTL genotype probabilities every 1 centimorgan across the genome. It'll use the genetic markers. If I have markers along the chromosome like that, it'll calculate the probabilities at those genetic markers, but also it'll overlay a grid 1 centimorgan apart all the way along. And it'll calculate for each individual QTL genotype probabilities at the genetic markers and at all these little spots in between we call pseudomarkers. And the function will calculate those probabilities and shove them into that object and then spit out the whole cross object. It'll spit out the whole cross object again with those genotype probabilities in there in a special way. Yeah.
If your marker density is always smaller than your step size, does interval mapping become a marker regression? If you never need to infer, I guess, a genotype between markers?
This will, if I had markers like every 10th of a centimorgan, it will still try to overlay another grid there as well. And unless you have complete data at the markers, and if you'll want to calculate these genotype probabilities anyway. This will also, another argument you can give is error.prob, which is a genotyping error rate. So I think that the default is really close to 0 or effectively 0, but you can give a somewhat higher genotype error rate, and this will smooth over the presence of errors. So if you have really dense markers, I guess I would consider leaving this step equals 1 off. The default step equals 0 is just do the calculation at the markers, no pseudomarkers added. In the arguments of these functions, you often don't have to write the whole name. So rather than write error.prob spelled out, I'll often just put if there's only one argument that starts with then it will know that. And I like to save typing as much as possible. Another thing, I don't know if you noticed, but in our studio, the up and down arrows will go back through the history of commands you typed. So you can go back to the previous one and then edit it if you want to add equals 0.01. But the key thing is that this calc.genoprob function, it takes as input the cross object, and it calculates these QTL genotype probabilities and shoves them inside in some way that I will explain later if you force me to. and it spits out the whole cross object with that thing again. So the usual way that I use this function is to assign the result of it back to the object itself. I could, assign it to something else. I mean, I can give it any name I want, but it will just mean I'll now have two copies of this QTL cross object, my whole data set, which in this case is not very big, but in other cases it might be huge, and one of which has these probabilities assuming the default value for the error rate, and the other with this error rate of 0.01. Okay? And the next function to know is called scan1, and that does QTL analysis with this single QTL model. I don't know why I put error there, SUG. So scan1, give it the cross object. It will do the genome scan by interval mapping. The default is to use the EM algorithm and calculate the LOD score at every position across the genome and spit out an object that contains that set of LOD scores. The function calc_genoprob I need to do first to calculate these QTL genotype probabilities. I assign it back to the cross object. Scan1 will do the actual interval mapping across the genome at positions that are determined by what I did in this step here. And what it spits out is an object that contains the set of LOD scores across the genome. And if I type plot out, it will plot the set of LOD curves across the genome. I'll say for this phenotype, I have a QTL on chromosome 7 and another QTL on chromosome 15. Yeah?
How does RCPL calculate the threshold to declare significant?
I recommend doing a permutation test. So we'll do that next. There's the function iPlotScan1 in QTL charts. You give it the output of scan1, and it will produce an interactive version of that plot of the LOD scores. It's not terribly interestingly interactive. I mean, it doesn't give you very much, right? I mean, here, it'll just, if you highlight over a marker, it'll tell you the marker name and the LOD score at that position. But if you do iPlotScan1 just with the output of scan1, it'll make a picture with just the LOD curves that when you highlight marker positions, it'll show you the marker name and the LOD score at that marker. If you click on it, it has a bit of gratuitous animation. I mean, completely gratuitous. If you do iPlotScan1, giving it both the output of scan1 and the cross object, now it will know about the QTL phenotype relationships. And it will produce the LOD curves connected to estimated effects at the QTL, where if you click on a marker, you'll get some confidence intervals for the estimated effects. And okay, but the key thing is to do QTL analysis, you first do calc_genoprob, and then you run scan 1. In scan 1, the default is to do the default is to use this EM algorithm with interval mapping. Yeah.
Can you write these out to a file, like an HTML file, or?
Let's see. You can press zoom and zoom it in bigger. You can get a snapshot of it. I think that if you if you assign it to something, there's a the question was, can I save this thing to a file so I can look at it later and interact with it? There is a way. I'll tell you later.
Can we have individual figure of
Say that again. Get the in.
Can we have individual figure of?
Get these individual figures.
No, for each KPL. Like one caption just one KPL.
Just one chromosome.
Yeah, one chromosome.
In the kind of the simpler plot, if you give it a chromosome number, it'll plot just that chromosome. And if you give it a vector, if there's a chromosome argument to this plotting of the scan 1 output, if you give it a chromosome or you give it a pair of chromosomes, it'll plot just those chromosomes. And the interactive plot will do the same thing. If I use this chromosome argument and tell it a pair of chromosomes, it'll show just those 2 chromosomes. Let's take a bit of a break.